Hello students, this is Dr. Faizan Mirza. In this video, I'm covering with you the ultrastructure of a plant cell when viewed using an electron microscope. And in this video, I'll be discussing with you the plant structures in detail, the chloroplast, vacuole, and cell wall. So let's start. In the plant cell, we start from the nucleus. You see the same four structures that you were discussing in an animal cell as well. The only difference being that now the nucleus is pushed to a side. We see the nucleus being pushed to a side in a plant cell in under the light microscope as well. The chloroplast can be seen, and these chloroplasts now have a double membrane. And actually, they had double membrane previously as well, but in light microscope this was not visible. In electron microscope it is visible. So chloroplast, mitochondria, and nucleus. These are the three structures that have double membrane. Apart from this, there is no structure within the cell that may have a double membrane. And these are grana. This was also visible in the light microscope. Golgi vesicles and Golgi body can be seen: cytoplasm, chloroplast, middle lamella, plasmodesma, cell wall of neighboring cell, vacuole having chloroplast and cell zap, mitochondria, smooth ER, rough ER, ribosomes, the cell surface membrane pressed against the cell wall, microtubules, nothing new. Everything that we have discussed in the plant cell, in the animal cell is there, and some additional structures are there as well. Let's move on. And this is a grayscale diagram in which might be asked to see to label and identify what structures can you see in the plant cell. Moving to chloroplast. Chloroplast is a double membrane bud organelle. The diameter is about 3 to 10 micrometer, which means it's larger than mitochondria. It carries out photosynthesis in green plants. Chlorophyll is present on thylakoids. What are thylakoids? Thylakoid is a new term for you. What's our thylakoid? Thylakoids are flattened membrane-bound fluid-filled sac present inside the chloroplast. And these are the site for the light-dependent reaction, which means the photopigment, which is your chlorophyll. It is present on the thylakoid membrane. What are thylakoids? If you look at this structure, this is the chloroplast shown to you. All these structures present inside the chloroplast are these thylakoids. These are all thylakoids. These singular structures, these are called thylakoids. And when we discuss the thylakoids stacked on one top of the other, many thylakoids present one on top of the other is called as granum. So granum and thylakoid simply are the same thing. It's just granum is many thylakoids present one on top of the other. Apart from this, inside the chloroplast, we have starch grains as well. We have lipid droplets, and there's a fluid film present inside the chloroplast, and we call that fluid as stroma. Stroma is present in the chloroplast. Light-dependent reaction occurs. The light-dependent reaction occurs on the thylakoid, whereas the light-independent reaction of photosynthesis occurs in the stroma. The circular DNA will be present there, similar to mitochondria. It's also having 70S ribosome, pretty much similar to mitochondria, and they can carry out their own protein synthesis. Moving on to cell wall. What's a cell wall? Cell wall in a plant cell is fully permeable. That's the first fact that you need to recall from your O level. Cell wall is fully permeable. So if a cell is placed in any solution, all the solution, irrespective of whatever the solution is, it will just trespass the cell wall and reach the cell membrane. But at the cell membrane, it will not be allowed to enter unless the cell membrane allows it to enter, because the cell membrane is then selectively permeable or partially permeable. So cell wall provides no barrier for or against anything moving into the cell. So anything and everything just can cross cell wall. Yes, the cell surface membrane will then make sure what goes into the cytoplasm or what comes out. Cell wall. Initially, the cell wall is formed around the cell. It's a primary cell wall. As the cellulose fibers are laid down, they all are parallel to one another. So this is how the cellulose fibers are laid down parallel to one another. This is how a cell wall is formed. This is called as a primary cell wall. Later on, sometime in the life of that plant, a secondary cell wall is laid down, where again cellulose fibers will be laid down, but they will be at a different angle to the previous one, but still parallel to one another. This is called as cross-ply structure of cellulose cell wall. This will be at different angle. It actually provides a good, you can say, resilience against the shearing force, or it just provides a structural support to the plant cells so they do not just burst open in any direction. So this provides a good strength. Mixture of other proteins and hemicellulose are present as well. Cellulose are relatively large structure, rigid structure having a high tensile strength. In xylem, we have lignin attached to the cell wall as well. Not only in xylem, in plants we study a tissue called sclerenchyma, and sclerenchyma also is having lignin in its cell wall. Apart from these two structures, there are no plant cells where lignin is added to the cell wall. Otherwise, the cell wall is just made up of cellulose, hemicellulose, and some proteins. Lignin adds the compressional strength to the cell wall so the xylem vessels do not buckle under the weight of water. What are the functions of the cell wall? Mechanical strength, prevent bursting, orients the cellulose fibers, and determine the shape of the cell. Adjacent cell walls of the plants, they form an interconnected pathway which is called the apoplastic pathway. We discuss this detail later on when we discuss transport in plants. So these parts of the structural detail, the functional details of the cell wall can be skipped for now. But when you are doing the chapter of transport in plants, you will already cover all this in detail. So then you can refer back to the structure of cell wall and add these points to your knowledge. You already will have covered this by then. So let's move on to vacuoles. In plants, vacuoles have the plants have a large central vacuole bound by tonoplast. Vacuoles contain a concentrated sucrose solution and other compounds stored as well. Turgor pressures build up and it allows the plants to grow in size. It elongates, and as it elongates by absorbing more water, the cell elongates as well. Vacuoles can also store some secondary metabolites that give color to the fruits or the flowers. Apart from this, vacuoles in a plant cells, they work similar to lysosome. They have hydrolytic enzymes as well. Mineral iron and protein sucrose can also be stored. Waste such as certain crystals of compounds are there in vacuole. Latex, for example, rubber is an example of these compounds present inside the vacuole, which we make for our use. Which actually we make use of. So if this table here basically just compares one against the other, your plant cell and the animal cell. What features do we have here? If you see plant cells have these and animal cells have these, you can just go through. There's nothing new here. We have covered everything regarding plants and plant cells and animal cells, and there's nothing new in this table. So that pretty much covers up the structural detail of the plant cells. So I'll just stop here, and this was all from my side regarding this plant cell detail. Thank you so much.